So we're talking about cell division, and we're still in the early part of the notes now because we want to talk about a little bit about eukaryotes. So in our last video, we looked at prokaryotes and how they replicate their cells. Now we're going to look at eukaryotes and how they replicate their cells. But before we do that, we should look at eukaryotes in general. We've done this a million times in class, and here's just a quick review. So remember, eukaryotes are animals. That means they don't have a cell wall and they can move, at least at some point in their life. Plants, they have cell walls and they do photosynthesis. Fungi are also eukaryotes. They have cell walls and they're heterotrophs. They are decomposers. And then protists, these are it's kind of a catch-all group, but it's any kind of eukaryote that's single-celled, or if it has more than one cell, its cells aren't specialized. So there's lots and lots and lots of different types of protists. We're talking about eukaryotes, but we're just going to compare that for a second with prokaryotes so you know the difference. Prokaryotes are bacteria, and so that means they're simple. They have no nucleus, they have no membranous organelles, and they're much smaller. Their cells are about 1/10 of the size of the cells of any of these eukaryotes. So more about eukaryotes. Eukaryotic cells contain a nucleus and membrane-bound organelles. So membranous organelles are things like mitochondria and vesicles and ER, endoplasmic reticulum, and Golgi bodies, and all that stuff. So they're organelles made out of membranes. Before cells divide, what needs to happen to their DNA? So you've got a cell, and we'll make it a eukaryotic cell. It's got a nucleus. There's its DNA. When you go from one cell to two cells, that's what this unit's going to be about, how you get from one cell to two cells. So we're going to talk in detail about, excuse me, we're going to talk in detail about how you separate out the chromosomes or the DNA. But this is just a question about what happens before the cell is able to do this. What happens to the DNA when you have a single cell and you need to go to two cells? This is way before you separate out the DNA. So the cells have to copy or replicate or reproduce their DNA. So if this is your chromosome, you've got to double it so that when you do mitosis, you're going to separate that out and end up with two chromosomes. So you've got one chromosome, there's one chromosome but it's duplicated, and then you do mitosis here where you're separating those out into different cells. So the instructions for making cell parts and making everything in your body are encoded in the DNA. So each new cell must get a complete set of the DNA molecules. After mitosis, each new cell will then have an identical copy of DNA. So here's your original cell. And once you do mitosis, you should have two cells with exactly the same instructions in them. So that means you just have to copy your DNA first. Make sure you do it right. So there's DNA, and you've got to copy your DNA. So you'll get two identical copies of DNA. So when we finish this unit, that's, I think, the next unit. We'll talk in detail about how you go from 1 to 2, but we're not going to do much on that right now. Here's what eukaryotic chromosomes look like. These happen to all be duplicated. They don't have to be duplicated. They can exist in a single form too, in which case they'd look a little bit more like this. But that's a chromosome, and this is a chromosome, and that's a chromosome. And this one, it's just hard to see the angle, but that's not one single blob. It's separated. So all eukaryotic cells store genetic information in chromosomes. So you can think about it as having 46 books, and here's one of them, and here's another, and here's another, and here's another. They're all different instructions. A chromosome, I think we've defined this before. The chromosome is made out of a DNA molecule and its associated proteins. Remember, we called those proteins histone proteins. Human body cells have 46 chromosomes, and sometimes we talk about them as 23 pairs because you got 23 of them from mom and 23 of them from dad, and that equals 46. Whoops, sorry. The number of chromosomes that an organism has is not related to its complexity. So kids are always kind of blown away by this chart. So penicillium has 1 to 4 chromosomes. Okay, whatever. This yeast has 18. A mosquito has 6. Okay, that makes sense. A housefly has 12. Garden peas have 14. Corn has 20. Adler's tongue fern. Okay, this is a plant. 1,262. That's just weird. Frogs 26, humans 46, orangutans 48, dogs 78. It doesn't mean that a dog is smarter than a human. It just means that they have more chromosomes. That's all it means. Notice they're all even numbers because they come in pairs except here. How chromosomes look when the cell is not dividing. So usually your DNA is uncoiled, and the reason you need it uncoiled is because you have to read it. You can have an enzyme that can come over here and read that little tiny section of DNA. If it were all coiled up, then none of your DNA would be used and the cell would die. So chromosomes can't be seen when cells aren't dividing. So when a cell is just hanging out doing the things cells do, it's all spread out. So the chromosomes are spread out or decondensed. Since the DNA is spread out, the instructions can be read to make RNA. So your body is mainly made out of proteins, and the way you get your proteins is that DNA makes RNA, and RNA makes proteins. And so since your body is mainly made out of proteins, and the things that proteins make, right? Some of the proteins are enzymes, and enzymes can put together carbs and lipids and all the other stuff. So you're mainly made out of proteins, and if you need proteins made in your cells, which you always do, then you're going to make them from RNA, which is made from DNA. If your DNA is all coiled up tightly like this, you're not going to be able to read it, and you won't be able to do any of that. So the only time you coil up your chromosomes is when you're going to separate them out. So here's what your DNA looks like. Here's the double-stranded double helix of DNA, and it's usually wound around these histone proteins to make these things called nucleosomes. You don't really need to know that term. And then these things wind when it's time to, this is what they're normally like when they're all spread out. When the cell divides, it's got to separate out its DNA, and if it were all spread out like this, it would be tangled and very delicate. You'd probably break some of the DNA. So the DNA starts to coil. And then it coils more, and then it coils more, and finally it's all the way condensed to this kind of chromosome you're used to looking at. So how chromosomes look when the cell is dividing? They look like that. When cells are dividing, the DNA is wound tightly around its proteins, the histones. The chromosomes become very compact so that they can be separated safely and accurately. So here's a duplicated chromosome, easy enough to split it right here and send this one to one side and that one to the other side. If this were part of a duplicated chromosome, how would you know which one to take where? And as you pulled it, it would probably break anyway, so that would be bad. Okay, have an awesome video. You'll have to wait for class to see that. Chromosome definitions. This is an unduplicated chromosome, and this is a duplicated chromosome. So you absolutely have to memorize this as being just one chromosome. And this is also one chromosome. The only difference is that this is unduplicated and that's duplicated. So as the cell goes through this thing called the cell cycle, when it first starts, when you just do mitosis and you get a baby cell, you got this. And the cell can be like that for a long time. And then at some point it will duplicate its DNA and then hang out a little bit longer, and then it will separate the DNA like that and send one to one new cell and one to the other new cell. And that will happen to all of the chromosomes. So this thing, the duplication, has to happen before cell division. So this duplication is not a phase of mitosis, it's what happens ahead of time. Chromosome definitions. This is one chromosome. This is also, whoops, this one over here is also just one chromosome, but it's duplicated. Each part of a duplicated chromosome, so a new definition here, is called a chromatid. So here's a chromatid, and this thing is another chromatid. So they're called sister chromatids. Identical sister chromatids. So the whole thing is a chromosome. Each part of it is a sister chromatid. If you only have one chromosome, you don't call it a chromatid because you just call them chromatids when they're part of a duplicated chromosome. And those are your identical sister chromatids. Chromatids are held together at the centromere. So this area is called the centromere, and they are still joined together here. So that's why it's one chromosome. Chromosome definition, centromere. This is the region of DNA right here where two identical sister chromatids come in contact. So the area is the centromere. But the actual protein that holds them together is called the kinetochore. And this is the thing that spindle fibers, these other proteins, attach to when you separate out chromosomes. Oops, here it is, kinetochore. This is a protein structure on the chromosomes where spindle fibers, or microtubules, remember the cytoskeleton, that's these things, where they attach during cell division to pull the chromosomes apart. So again, I use kinetochore and centromere pretty interchangeably. Centromere is the region and kinetochore is the physical protein structure. Karyotype. Remember we learned the words prokaryote and eukaryote, and prokaryote meant before nucleus and eukaryote meant true nucleus. So karyote has something to do with nucleus. So karyotype has something to do with the stuff that's in a nucleus. So this is a picture of the chromosomes from a cell arranged in pairs by size. So here is a karyotype, and you can look at this and say, okay, here's chromosome pair number 1. This one came from mom. That one came from dad. Here's pair number 2. Remember we said there's 23 pairs. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, blah, blah, blah. 22. Here's the 23rd pair, the X and the Y. They're a little bit different from each other. But those are the 23 pairs or the 46 chromosomes. So I can tell this is probably a human because it's got 46 chromosomes. You can tell that this is, they order them in size, so number 1 is the biggest one. And they come, it's really important to understand that they come in pairs because you get one from your mom and one from your dad. So this is not exactly the same as that. This might code for blue eyes and that might code for brown eyes. And this might code for wide hands and that might code for narrow hands. And there's a million different genes on here, right? And this one might code for a pointy nose and that one might code for a wide nose, and that over here might code for a long nose versus a short nose. So you have all these different traits, and these code for the same kinds of things, but not exactly identical things. And then you'll notice X and Y are not the same shape, and that's kind of interesting stuff. So if you have an X and a Y, you can tell that this individual, this karyotype, came from a boy. If there were 2 X chromosomes, you'd know that they came from a girl. So the first 22 pairs, more definitions. There's a lot of definitions in this unit. The first 22 pairs are called autosomes. So everything, ooh, I need a different color, don't I? So all of these, pretend I drew that around. All these are autosomes. And so an autosome is anything that's not a sex chromosome, and these are the sex chromosomes. So the last pair are called sex chromosomes. Blah. So XX is for a female and XY is for a male. So I can tell this is a male because it's got a big chromosome and a little chromosome. So this one is a boy because it's got an X, and then this teeny little one is a Y. It's not shaped like a Y. It's just little, and so you can't really see the separation here, but it exists. And here's the bigger X chromosome. So the Y chromosome decides whether it's a boy or a girl, and that's the Y chromosome. And that's the X chromosome. And so I have all the DNA that I need, almost all the DNA I need to be a boy, but it's not turned on because I don't have a Y chromosome. So I'm missing a little bit of DNA, this teensy little chromosome here. And there's a master switch on here called the SRY gene. And that SRY gene is the boy gene. If you have that, it's a master switch, and it turns on a whole bunch of other DNA that's in a whole bunch of other chromosomes, and it determines boyness. So if you don't have that SRY gene, then you are not a boy. Why do the chromosomes come in pairs? Because you get one set of instructions from your mom. Thanks, Mom. and one set of instructions from your dad. Thanks, Dad. So while we're talking about karyotypes, let's take a quick look at this one. If you look at this one, you'll see there are 48 chromosomes. So here's pair 23, and then here's the X. Oh, this has got to be a girl chimpanzee. I think it's, yeah, it's the same thing in them. So that one has 23, 24 pairs, so that's 48 chromosomes. So that's not a people one, that's actually a chimpanzee one. Whoops. Here's a mouse karyotype. There are 20, whoops, I messed this up. There's 20 pairs. There are 20 pairs of chromosomes. If you can fix that in your notes, that would be good. 1, 2, 3, 4, 5, 10, 15, 20. So 20 pairs, that means there's actually 40 chromosomes, right? So I'm just showing you this so that you get the idea that their karyotype is going to look different from your type. And so you can tell it's not a human just based on this. But notice that they're all in pairs because mice have a mom and a dad too. All right, and I'm going to stop here.